Hello. Um, my name is Sia Garg, and today it is May 23rd, 2023. Um, okay. So today I'm going to be talking about anatomy and physiology of the retina. So the first thing I need to talk about is just kind of like some anatomy of the eye itself, because the retina cannot work unless other parts of the eye are working as well. So just to talk about the retina itself, the retina is a light sensitive surface where the light gets focused onto, and it will transfer the neural signals into electrical signals. And a unique characteristic of it is that it contains photoreceptors, which allow for this to happen. And then it needs to coordinate with other structures of the eye. So the first place where the light shines onto is the cornea, and it is a transparent sheet of tissue where the light will enter, and the light will bend only slightly here, and then it will go to the lens, and this is where the light will bend a lot, and this is a transparent disc which really focuses the light, and it will focus it onto a part of the retina called the fovea, which I'll talk about later. Then we have the pupil, which is that dark spot seen over there, and it is in the center of your iris. So the iris is a muscle that allows for the pupil to open and close, allowing different amounts of light to enter, and the iris does this by contracting or relaxing. So, talking about the anatomy of the retina, there's five types of main neurons in the retina. So, the first one is called the photoreceptor, and this is what allows the light energy to turn into electrical signals, which is what I said before. And it is on the bottom layer, and it is touching the epithelial layer, which I'll talk about a little bit later on. So, the two types of photoreceptors are rods and cones. Rods are better suited for low light, so during the night, and then cones are higher acuity vision, so they're better for the daytime. There's more rods in our retina than cones. However, cones are concentrated in the fovea. There's no rods in the fovea, and then rods are also more sensitive to light. The next thing we're going to talk about is the bipolar cells within the retina. So there's two types. There's light on and light off. So this is also called off center and on center, and this is controlled by the glutamate release based off the rods. So, for example, if the rods are turned off, then the light it would be light on, and then there'll be low glutamate release, and then vice versa. Ganglion cells um, are what the electrical signal will go from the bipolar cells to the ganglion cells, and it's actually the axon of the ganglion cell where the optic nerve forms. So just to give a little context, the optic nerve is what allows for the electrical signal to go from the eye to the brain, and the optic nerve where it enters the eye, that is called our blind spot. So that's kind of like an area where you can't really uh, see anything. And so the ganglion cell is the surface of the retina, and it's associated with only a few receptors. So when they're towards the edge of the retina, where there's a lot of receptors associated with one ganglion, the vision is not as sharp. So it is better for a ganglion to only be associated with a few receptors. So then we have horizon cells, and this mediates a lateral inhibition in the retina. And then we have amacrine cells, and this essentially facilitates the flow of the electrical signal from the bipolar cells to the ganglion cells. So the optic disc is within the photoreceptor, so it's within rods and cones, and so it contains different proteins. The special one in rods is called rhodopsin, and in cones it's called photospin. Sorry if my pronunciation is not good. The fovea is a little bump in the retina, and it is it's surrounded by a muscle called the macula, and together this area of the retina is has the most acute vision, and it's the center of the visual field. Um, and this is the one where it has the highest resolution. The reason for that is because it does not have any axons. The periphery of the retina, where there's a lot of rods, there's a lot of bundles of axons, and because of this, when the light ray enters the retina, it has to go through um, those bundles of axons before it reaches the rods, and because of this, sometimes energy can be lost. This is not the case in the fovea, however. Then is the choroid, which is a dark pigmented epithelial layer, and it touches the photoreceptors. And so it essentially absorbs any of the light energy that escapes the photoreceptors, just to prevent any distractions or distortion of the visual image. And the reason why it's dark is because it contains melanin, and it also helps to nourish the retina overall. So the retina is associated with our vision. And so vision is essentially when light reflected from an object and in the environment is translated into a mental image, and that mental image is formed within our brain. So it's very important for there to be a clear communication between the eye and the brain. So, how this happens is it's essentially a cascade. So, the light rays will enter the eye. And then once it goes through the cornea, the lens, it will reach the retina. In the retina, it will hit the rods. And some of them will hit the rhodopsin, which is in the internal discs within the rods and cones. In the cones, it's called photopsin, but in rods, it's called rhodopsin, and it will essentially change the conformation of a molecule within the rhodopsin called retinol, and it will go from a cis to trans conformation. So essentially it goes from a bent to a straight conformation, and this will also cause the rhodopsin to also change shapes. Then there's a molecule called transducin, which contains alpha, beta, and gamma sections. And this was originally attached to the rhodopsin, but when that conformational change occurs, it will leave the rhodopsin. And then the alpha subunit of the transducin will attach to PDE, which is a complex that converts the cGMP to GMP. So what this means is that cGMP, when it's bound to sodium ion channels, sodium can enter the neuron. However, when it converts to GMP, the sodium channels will turn off, and due to this, sodium cannot enter the cell. When this happens, hyperpolarization will occur, and this will just turn the rod off. So just to summarize, light will enter, it will hit the rod, it will go from being on to off. When this occurs, on center bipolar cells will turn on and be activated. And then in turn, um, with the help of the amacrine cells, the off on center ganglion cells will also get turned on. So it will go from bipolar to ganglion cells. Then the cells will send the electrical signal to the optic nerve, which will enter the brain. So that's essentially what you need to know when it comes to the retina itself. Once it gets to the brain, the optic nerve from the retina will go to the optic chiasm within the brain, and crossing over may occur. So what this essentially means is there's a nasal side of the eye and then the temporal side. So any of the rays that entered from the nasal side, those will cross in the chiasm, but the temporal side, they will not cross. So there's a mixture essentially. And so the visual neurons will synapse after that in the lateral geniculate body in the thalamus, which is located right here. And then it will terminate in the occipital lobe. And this is within the visual cortex, and this is how the visual is made within the brain. Something that's a little interesting, however, is that there'll be some collateral pathways which will go from the thalamus to the midbrain, and this is where they're going to synapse with efferent neurons of the cranial nerve 3, and this will in turn control the diameter of the pupils. And so this is associated with something called consensual reflex, and this is something tested within when you go for a eye doctor appointment. where they will take a light and they'll point it to your eye, and they're just checking to see if your pupil will change sizes. So they want to see if it will constrict, right? Because if there's a bright light, then your pupil should constrict, because if too much light enters your retina, it can be dangerous. So that's something that is just really interesting, as it can show that there is some sort of connection between our brain and our eye, and communication and coordination. And that is pretty much it. And then I made a worksheet in order to just kind of summarize everything that we learned about the anatomy and physiology of the retina. And all of the answers of this can be found within the slides itself. There will also be an answer key that will be shared. Thank you.